So my talk is a bit general, that I wanted to talk a little bit about AAV gene therapy and to follow Dr. Bhattacharya that already showed a nice, lovely slide. You can think of gene therapy interventions nowadays in two ways. They come, I suppose, in two flavors. One in which patient cells are taken out of the patient. They're taken into cell culture and to the lab. They're genetically modified, and that's evolving very rapidly how they are engineered. We started by using viruses, but with the advent of CRISPR technology, we can actually now correct the genes directly and don't have to use necessarily viruses to do so. So actually, that's the ultimate goal of gene therapy, is to really correct the genetic defect or mutations that are in the genes that cause the diseases. So that technology is there right now. And ex vivo really is ideal for that technology, although there's quite a few trials now ongoing in human as well, in vivo. So that you modify the cells and then you put them back into the patient. This is a technique that is used for a lot of hematological diseases. So you modify the bone marrow, you transplant the cells back into the patient, and hopefully you correct the problem with the hematological or with the blood. The other approach, of course, is actually direct delivery of DNA. The most effective one is really using viruses. Understandable, because viruses have evolved to actually infect cells and deliver their genes to cells so they can replicate. But of course, we do not have our viruses replicate. The last thing we want to do is substitute one problem by another. So we obviously engineer the viruses to not carry their own genes. And then they're infused back into the patient. Although I have to say that, this yellowish kind of AAV or vector, I would be very reticent to put it in patients, because really the AAV really has absolutely no color, or viruses really don't have much color. So that color there is a little suspect. But anyway, so what I'm going to talk today, and that the previous speaker spoke about, is the in vivo gene therapy, and particularly using adeno-associated viruses. And it's really an interesting story, because it really is the story from a very humble start as a contaminant to being the preeminent vector for in vivo gene therapy, and really to be able to treat genetic disease, and some of them actually with approved products. Now, this doesn't take some time. This was discovered in 1965 as a contaminant of adenoviruses. And so you can see here that the adenovirus in a much, much, much smaller AAV. And so because it was found as an adenovirus contaminant or associated with it, it became known as an adeno-associated virus. And of course, since that 1965, we now understand the structure of the protein in great detail. I mean, we actually know where every atom that makes this capsid is located. And, not only we understand that, but we actually really start to understand the function of different areas of the capsid. And you can see here some labels of important regions of the capsid. For instance, this little hole in the capsid here, that it looks like a pentagon. There's a little hole in here, and that's where the viral genome is inserted to. And we also know that, for instance, in this area here that has these little mountains, this is actually where the viruses interact with cells to infect them. And we've learned this through the work of some giants in the field, and Avis Badanji McKenna was a giant in the field that contributed immensely to our understanding of the structure and the function of AAVs. And unfortunately, we lost them to ALS this year on March 3rd, which of course we all miss her immensely and her scientific contributions. So AAV, adeno-associated virus, it's, people joke actually that this is almost a virus. That's what AAV means, because it really is a very simple one. It has two genes. That's it. That's all that makes this virus. And it only makes 8 proteins. Of course, all of them very important for its function. It's you can think of it as a nanoparticle, and it's about 20 to 25 nanometers in size. It has a single-stranded DNA genome. And because it's such a small capsid or such a small virus, you really can't fit a lot of DNA inside. We have our playroom, or our little sandbox is really pretty small at 4.7 kb. So you can fit inside 4,700 base pairs of information. That's it. Maybe sometimes you can stretch it a little bit more than that, but that's all you can play with. And the good thing about this virus, and part of the reason they're part of this dependovirus genus, is because they depend on another viruses for their replication, such as adenovirus or herpes simplex virus. Now, this is just a timeline, and I admit to it that this is a bit parochial because there are some investigators here that are at UMass Medical School, but from the beginnings of 1965, of its first discovery, took really an enormous amount of work to get to the first time in 1980 when people started being able to clone these virus genes or genomes to understand what makes them, right? And so each step here that you can see here in the timeline, sort of these milestones, and they really correspond to technological developments that have occurred. So in 1980, recombinant DNA came out, started to become very popular, and started to be used. That's why we're able to clone the genome. In the early 90s, we started to know how to make these viruses for clinical application. In fact, the first human trial for cystic fibrosis done by Terry Flott while he was at Johns Hopkins Medical School. He's now the dean of the UMass Chan Medical School. And then around mid 2000s, or well, I guess early 2000s, I suppose, very early, Guangping Gao, while he was at Penn at University of Pennsylvania, he actually discovered really important new capsids, such as AAV9, that have become the mainstay of a lot of the clinical translation that is ongoing right now. In 2012, it was the first product in the world approved, was in Europe, Glybera. Unfortunately, that product really was not adopted because there was questions about its efficacy, and since then it has been removed from the market. 2017, Luxturna was approved in the US. In 2019, Zolgensma for spinal muscular atrophy was approved. So you can see that from 1965, took some time to get to the first approved drugs through enormous technological developments and seminal findings. Some of them are here in the form of the titles of the papers. So it does take some time from the early phase and someone actually caring about almost a virus to actually become something extremely important for treating genetic diseases. So these are the two drugs that are approved presently in the United States. One is to treat a genetic blindness, Leber's congenital amaurosis here, RPE6 mutations in the RPE65 gene. This is really transformative because patients that are unable to see eventually, once they are treated, they start to be able to recover their vision. So really is a transformative treatment. And the AAV9 for spinal muscular atrophy, a disease that is colloquially known as the floppy baby disease because they have no control over their muscles because spinal motor neurons die. The results with this drug really have been transformative, although they do have side effects. But the fact of the matter is that patients treated with this AAV gene therapy, many of them are alive today. Actually, the majority of the patients that were treated are alive today versus actually dying pretty early on, and in the most severe forms, dying actually before 1 year of age. So AAV, during those 50 years of science, one of the things that we've learned how to do is gut the house and make new rooms inside. So we took out the genes. So the two genes that I talked to you about, called RAP and CAP, doesn't really matter their names. But essentially what we do in gene therapy is that we gut the virus. We keep just two elements, tiny, tiny little elements called ITRs. And we replace that with genetic information that colloquially we call transgene. And what it is it has the genetic information to make a normal protein that is missing in the cells. It has a genetic element that is necessary to tell the cell to make an RNA that gets then translated into the protein. It requires also a signal that tells the cell or the transcriptional machinery to stop making the RNA. So this is sort of the basic structure of all AAV vectors, and they come in different flavors. And what you have here on the left side is an electron microscopy. So this is really a picture of the viruses. And actually, what you can see here is empty capsids and full capsids. The empty one says the dark center in the middle, the full ones don't. And this is actually something that you always end up finding in your AAV preps. And in fact, nowadays, there is quite a bit of discussion of whether side effects, immunological side effects, are caused by having too much of these empty capsids in the AAV preparations. So why do we use AAVs? Well, first of all, because they don't carry any genes. So in terms of toxicity derived from viral replication of viral genes is not there. They are highly efficient for in vivo gene delivery. They do not integrate into the genome, and I put in there for the most part, because if you look carefully, there is a certain percentage that it does, but nonetheless, they have a very low genotoxic potential, meaning that after treatment, it's very unlikely that patients will develop tumors because of the treatment. The other aspect that we like them for is the fact that once you deliver them to cells that are not dividing rapidly, they can actually stay and express genes for a very, very long time. And that's why from the previous speaker, you saw that the notion right now is that a single time treatment will be enough to actually provide therapy long-term, potentially lifelong. What we know from animals is that really is lifelong. But of course, humans live a little longer than animals. Now, on the other hand, while in the past we used to say, well, the AAVs are non-immunogenic, then we started to say, well, they are not terribly immunogenic. And now the clinical trials that have gotten to very, very high doses actually have shown that humans tend to respond quite poorly to it, especially if you cross a threshold of about 10 to the 14 vector genomes per kilogram, which 10 to the 14 really is a mind-boggling number of amount of particles injected into a person. But what we have also found is that if we use immune suppression and immune modulation, we can actually tamp down those type of responses that are found in human and actually not quite in animals. And that's the difficulties. Modeling what happens in human in animals really is quite tricky. So, our knowledge and thoughts about AAV really have evolved in clinical trials. And our notion that they were not very immunogenic started from trials where we used very small amounts of virus in a human, and everything seemed fine. But as we need to treat diseases that are very widespread and we need very efficient gene delivery, we start to go up on the dose, and that's when we're starting to find some of these complications from AAV gene delivery in vivo. Now, how do we make them? During those 50 years, what we've learned to do is actually make them in a safe way, meaning that we still need the genes from the virus, that genes that I told you about, the wrap and cap. We also need the adenovirus genome, but we only need a few genes from them. So how do we do this nowadays? We actually make these circular pieces of DNA in bacteria. And what we do is that we then introduce them into human cells in cell culture. And so they have all the instructions necessary to make, not only make the AAV genome, but then assemble them properly into the AAV capsids without any other viral genes inside. And so this is essentially the mainstay of production of AAV vectors. And it's interesting when I talk to people about this is that gene therapy really is a very strange field in science, because the reality is that you don't see anything. I've shown people tubes of AAV that are worth quite a few $1,000s, and it looks like water. So everything that we see is at the microscopic scale, and we actually use all sorts of organisms to actually do the job for us. So bacteria make this DNA, then we have cells in culture making our virus. So it's actually interesting to think about it that we deal in the nanoscale environment where we really can see, and what we know is based on indirect observations and tests. So then after we make them in these cells, then it's a question of, can we purify? Can you clean them up really well so that when you put them in animals or when you put in human, we really have safety, right? That we don't cause a problem by bringing along other proteins, other pieces of DNA, RNA, and so on. So what we've learned over the years is really how to clean it up to make it almost pure or completely pure preparation that then we can infuse safely into animals and in patients. Now, one of the interesting things about AAVs that have been incredibly useful over the years is that once you have designed a vector that is shown up here in a schematic form, you can actually insert it into pretty much any capsid. And the work in the field started with AAV2 because it was the only one that was known. Once the AAV9 appeared, AAVRH10, you heard about AAV Myo. All you need to do is just introduce the genes that make these capsids when you put them in the human cells, and that's it. So we don't need to further modify this vector genome to actually use another capsid that has new properties and better properties. So that's an important, actually, property of AAV. And on top of them, they also come in different flavors. And when I say flavors, the genetic flavors here, I suppose. So I told you before that it's a single-stranded genome. So DNA is double-stranded, but these viruses actually make a single strand. But in addition to this form, which was the classic form that was used in most trials, you can actually do a genetic trick where you trick the virus into packaging these double-stranded genomes or self-complementary. The advantage of this type of design is that it's a lot more potent than this single-stranded, because when the virus enters the cells, it's ready to be expressed because it has these double-stranded genomes. So these are much, much more efficient than these versions. But of course, like almost anything, lunch doesn't come free. And because you're actually putting more DNA inside that is double-stranded, now that capacity that I had told you of 4,700 base pairs, you can only put now 2,400 base pairs. So about half of what you can do in this ones. But if you can fit it inside, these viruses really are much more potent than the single-stranded viruses. Now, in terms of developing therapies for muscular dystrophies, what are the challenges? I think the previous reviewers outlined them very nicely. Obviously, the muscle mass is distributed throughout the entire body. It's very, very large, which means that we need to use systemic gene delivery to hopefully alter the course of disease progression. That has implications. You've heard from the previous speaker, the toxicities that have been observed in liver and actually in the central nervous system in the dorsal root ganglia. And that's because, the liver really is the organ in your body that pretty much takes up everything and anything to detoxify it. So AAVs end up a large majority of it in the liver, and then they also get elsewhere. But as you heard before as well, there's efforts to engineer capsids that actually go very, very well to other tissues and not quite as much to liver. One of the big challenges that, again, you've heard earlier today is that in the adult population, about 50% of individuals have neutralizing antibodies to AAV, which makes it very, very difficult to treat patients that have that. And that's why there's this effort to actually screen the Indian population or the patient population in India that has for the presence of these neutralizing antibodies, because it's pretty important to know that, because let's say that all the population has antibodies, then we have to work on means by which we can remove them so that we can treat patients. Overall, when you start some of these clinical trials, especially that they are delivered into the bloodstream, so that's what I mean by systemic here, usually you have to select patients that don't have too much of these neutralizing antibodies, because otherwise it does not work at all. It's actually a waste of the virus. You might as well just throw it out because it doesn't have any effect. So this is actually a very critical type of activity to understand in the patient population what is the prevalence of neutralizing antibodies. One of the things that we're quite fortunate is that I mentioned before, the discovery of AAV9. The first thing that AAV9 became famous for is its ability to cross the blood-brain barrier. But it turns out that it can actually mediate gene transfer to muscle at extremely high efficiency. And what you have here is monkeys that were treated at different ages, at 1 day of age, 30 days of age, 90 days of age, with an AAV vector expressing a green fluorescent protein. And you can see here a monkey that was injected with vehicle, meaning phosphate buffer saline, where the vectors are made or formulated. there is no presence of the green fluorescent protein. And if you look throughout multiple muscle groups throughout the body, you can see extremely high efficiency gene transfer to many, if not all, types of muscles that have been analyzed. So obviously, AAV9 has become one of the vectors that is being used for gene transfer to muscle. Another one is AAV RH74. There's this company in Cambridge, Massachusetts called Sarepta Therapeutics that is developing a gene therapy for Duchenne muscular dystrophy. And you can see here the degree of gene transfer. So this is using an antibody to detect expression of this microdystrophin that they're using, so which is the smaller version of the protein. You can see in the same patient before treatment and after treatment, and it's very, very clear that the protein is being expressed in a considerable number of muscle cells or muscle fibers, suggesting that, in fact, we should have quite a significant therapeutic effect, although one could argue the rest of the afternoon or evening in your case about the results, whether it really is showing efficacy or not. Now, in terms of GNE myopathy, I'm sorry that I missed Sakra's talk earlier today, but the path towards the clinical trial is actually relatively straightforward. Where you start is obviously in vector engineering. You want to have some proof of concept in vitro. Actually, it should be in vivo, really. Ideally, what you have, if there is an animal model, is to demonstrate that you can change the course of the disease by your AAV treatment. And ultimately, what you need to do really is an in vivo safety. And there were questions about the safety to really demonstrate to the regulatory agencies that, in fact, we can put them into patients with some degree of knowledge about safety, and that more likely than not, the patients will be fine. Now, along with this, it's actually very, very important that clinical trial design starts, and that's why it's very important to actually interact early on with physicians that have experience with patients, following patients that have GNE myopathy. Having natural history studies is incredibly important because that can tell us how the disease normally progresses in patients from the onset of symptoms to, as the disease progresses. Because during a clinical trial, what we want to know is, do we change the course of disease progression? Obviously, if we cure a patient, well, I think all of us can understand that. But often that's also not where clinical trials start. And the previous speaker mentioned that, well, we have to find the right dose. And the fact of the matter is often you can only find the right dose in humans, because what we find in animals often does not translate into humans in terms of an exact translation of dosing. So usually a clinical trial is a dose escalation, also because of safety, because while you've done experiments in animals suggesting that the drug should be safe, well, as I've told you, humans, we tend to throw some surprises into the AAV gene therapy field. So you start cautiously. You start at doses that are a bit lower than the maximum dose where you want to get, and you progress up towards that dose where eventually you'll be hopefully fully therapeutic. So, one of the biggest things that is between some of these phase one, this initial part of proof of concept and getting to a clinical trial really is AAV manufacturing, which for systemic gene delivery really is a major endeavor, because the amounts of viruses that are necessary really are considerable. You've heard me saying that in some cases 1 × 10^14 vector genomes per kilogram. And if we're treating adults, well, you can do your own calculations and multiply that by the number of patients, and suddenly you find yourself in scales that are quite staggering. So one of the things that I wanted to bring up is, designing an AAV vector for GNE myopathy. So, well, obviously it's a myopathy. After all, that's its name. So that's obviously a muscle disease. But in my perspective, it's actually quite important to always understand, where is this gene expressed usually? Is it just in muscle, or is it expressed elsewhere as well? Turns out that I was very surprised when we spoke earlier this summer. Dr. Bhattacharya I thought it was a gene that was expressed exclusively in muscle cells, and I was very surprised to look at the Human Protein Atlas and really find out that these genes actually express in a very wide variety of organs, almost universally. And in fact, the highest amounts of RNA are not even expressed in the muscle. Turns out that liver has actually expresses very, very high levels of the RNA for GNE, for the GNE gene. And then the protein is expressed in different amounts in different tissues. And again, the skeletal muscle or the muscle is not even the one that expresses the highest amounts of this protein. So it's something that is important to consider in the design of the viral vectors and really understand what components of the disease need to be addressed or what organs need the gene transfer to achieve a therapeutic effect. Obviously, given that it's a myopathy, the first target that one thinks about really is the skeletal muscle. But I think it's important to take into account these other aspects of the expression profile of this gene in human. So just to give you a very brief, I apologize, I lost a little bit of track of time, but I'm going to try to go through this quite fast, of giving you sort of sense where gene therapy has been at UMass Medical School, now called UMass Chan Medical School. We have in 2008, we started two centers, Jorge Gene Therapy Center and the RNAi Therapeutics Institute, dedicated to two technologies that were emerging, which is therapies based on RNA and therapies based on using viral vectors to really power the development of new therapies for genetic diseases. And those two motors of innovation have really brought forward quite a lot of programs developing gene therapies, both in terms of AAV. There are about 46 programs, using oligonucleotides. There's about 39 programs in CRISPR. There's 6 and counting upwards very, very rapidly as well. So the question is that our translation from all this preclinical work and development work towards the clinic has been relatively small. I mean, we've conducted 5 clinical trials with AAV, 2 clinical trials, oligonucleotides, and so far none with CRISPR. So the question for us is, how do we make this transition from the discovery phase to the clinical translation? And this is just a summary of the investigators that have been involved in the different diseases that we've translated to the clinic. And there is a number of them, including ALS, where Bob Brown, his picture is right here, working with Chris Mueller and a colleague at Mass General Merit. They've done quite a few clinical trials in ALS, both in SOD1 caused ALS as well as other forms of ALS. And me and other team members would have developed and now in clinical trials, AAV gene therapies for Tay-Sachs disease and GM1 gangliosidosis. So these are some of the programs that have gone forward to clinical application, and they're now in clinical trials. But nonetheless, having done 5 clinical trials in AAV, 2 in oligonucleotides, we are left with quite a few other programs that we really need to move forward, or we'd like to move forward quickly. So this translation to the clinic is really the next phase of gene therapy at UMass. And for that, we've created sort of the third wheel of the puzzle, that is the Translational Institute for Molecular Therapeutics that was recently formed. And the mission is pretty simple: it's accelerate to translation to clinical trials of life-saving gene therapies. And it doesn't really matter if it's AAV based or oligonucleotide based. And what are our goals? Based on our experience of those 5 clinical trials that we've taken forward, and as mentioned by several of the previous speakers, is the cost of entry to clinical trials. As you move towards the clinic, it really makes the discovery phase look incredibly cheap, and certainly is not cheap. The discovery phase is not cheap by any stretch of the imagination. But when you move towards human application, the costs spiral almost out of control. And personally, I think they're right now, at least in the United States, they're absolutely out of control. So our goal really is try to analyze the different costs that are involved, production being a major one of them, and really try to figure out ways in which we can positively impact by reducing those costs of those activities. So our goal is to have end-to-end support after the proof of concept studies and increase the number of clinical trials conducted at UMass. This is sort of the notion behind the Translation Institute, is that, you know, we can start these programs from any point. Either UMass investigators that are interested in a particular disease for some reason, patients, foundations, biotech, pharma that come to us. Obviously, we need to figure out how to finance the development programs. And then the Institute sits here in the middle of having regulatory support, GMP manufacturing, and eventually the ability to also conduct IND-enabling tox studies. These two components here, the manufacturing, these IND-enabling toxicity studies, really are what makes it extremely difficult to go from the discovery to translation. But this is what we're putting in place right now. And ultimately, our goal is to go to clinical trials at UMass or before. And so this is just finished with this building right now. So a little cartoon here, watercolor, a cute watercolor of sort of the discovery buildings at UMass. This one here is not built yet. It's being built right now. This is a rendering from the architectural firm. This is the building where my lab is located right now. And this was actually the first the first building at UMass dedicated to research, aside from the buildings at the medical school. And what we're building in this new building on the, I think, 9th floor is actually a GMP facility where we can actually produce vectors to be used in clinical trials. So we're very hopeful that we can accelerate the transition from discovery to clinical trials, really to accelerate giving hope to patients that obviously need these treatments. And so it's wonderful to know that there is a very strong organization and impetus to do this in India for a disease that certainly affects lots of people, unfortunately, in India. And certainly, you know, we're here to help and collaborate, because honestly, there's way too many rare diseases in the world for us to be competing across different diseases, because honestly, there's 7,000 diseases. This is not something that a single country is going to be able to do it on its own. So I think it's really needs everyone to be invested in different diseases, and obviously ones that are more meaningful to a country. It's wonderful that a country like India has this endeavor, and this is a global effort. And so collaboration is actually critical for us to be able to accomplish this goal of treating patients that have no options nowadays.